For example, plants are very commonly polyploid, and polyploid means, poly means many, and ploid means how many copies of chromosomes you have. For example, watermelons and bananas, they're both seedless and they both have 3 copies of chromosomes, hence called triploid. Blackberries and peanuts have 4 copies, so are tetraploid. Sweet potatoes and wheat are hexaploid for their 6 copies, and strawberries can be up to octoploid. And there are advantages to being polyploid. For example, when fruits become polyploid, the actual fruit becomes bigger. And so you can see from a commercial standpoint, it would be advantageous for humans to want to take a plant, which originally wasn't polyploid, and turn it into more copies of chromosomes so that the fruit will become bigger and they can make more of a profit. So there's certainly a commercial value in polyploidy in plants. The question becomes, how do you make an organism polyploid if it wasn't already? And for that, we have to know about the cell cycle. So far, in the cell cycle that you were introduced to, the cell cycle starts at G1, and G1 just means growth. And on the side, I have a cell, and we're going to make it nice and easy. This is going to be a 3-chromosome cell. And as you can see, there are 2 copies of each chromosome. So each type of chromosome is different color-coded. There's 3 colors, so there's 3 chromosomes, and there's pairs of each. And so, because there are pairs, this is what we call 2N, or diploid. So we're going to start with a cell that is already diploid. It's not polyploid. Now, G1 stage is actually pretty unremarkable. All that happens is the cell's cytoplasm gets larger. It undergoes regular cellular metabolic activities. The cell just gets bigger. It's not until we get to the S stage, and S stands for synthesis. The synthesis stage of the cell is characterized by duplication of the DNA. Notice that the 2N cell, every copy has duplicated itself. So before there were 2, now there's 4 of each type. And so essentially, this has become 4N. And so we wouldn't call it diploid anymore, we would call it tetraploid. And normally this is actually temporary for the cell, because what's going to happen is it will undergo G2, which is, again, another growth cycle. G2 is the stage that comes just before the M stage, or mitosis. And what's supposed to happen in mitosis is we start off with our 4N tetraploid cell. During mitosis, the chromosomes line themselves up in the middle of the cell and they get pulled to either side. You'll want to look at the act of mitosis in depth, but for the purposes of this video, I will very much shorten mitosis. A line will kind of go right down the middle, and the chromosomes will be split apart, and the cell will divide itself into 2 daughter cells, and the 2 daughter cells will each have, again, 2 copies. So these 2 copies went over here, these 2 copies went over here, etc., etc. Just noticing my color coding, that thing went black. That should be blue. So what should end up happening is that your 2 daughter cells that are created, they go back to having 2 copies. So this is 2N and this is 2N. So they are both diploid. And that's what's supposed to happen normally. And so one of these daughter cells will just undergo its cell cycle again, etc., etc. And this always continues. We have to remember that mitosis is always, you are originating as a 2N or diploid cell, and you are eventually ending up as a diploid cell. Or if you started as a tetraploid cell, then you end up as a tetraploid cell. My point is that during mitosis, ploidy is maintained. Now, you're probably thinking, I thought we were trying to make a polyploid organism. You just showed me cells ending up back at 2n. Well, what we do is we disrupt the cell cycle. The cell cycle will continue, so we still let G1 occur. We still let S occur, so we duplicate the genetic material. But if we want to make a polyploid plant, then we would use what's called a mitotic inhibitor, something like colchicine. And if you prevent mitosis from occurring, then you never get a splitting, and these daughter cells wouldn't be created. And all you'd end up with, if we use colchicine, then the ploidy of the cell won't change. So the cell will undergo the cell cycle again. This cell is going to grow up to be a tetraploid plant. So 4n. And eventually, that tetraploid plant is going to want to make sperm and egg. That presents some interesting mating opportunities. And here I'll show you. If you start off with a tetraploid plant, the tetraploid plant, if it were to make sperm and egg, you'd basically split the chromosome number in half. And so you would make 2n and 2n sperm or egg. And the reason why that's important is because if we were to cross a tetraploid plant with a regular version, so a diploid plant, that's just going to be n, otherwise known as haploid. So these guys are just going to put haploid. Or you can kind of think of it as 1n, or one copy. So if we made sperm and egg from just the regular one, then we could take a 2n egg and a 1n sperm, and we could combine them into a zygote. And the zygote would end up being 3n. Dot plants that would grow up to be 3n. What's interesting is that in plants, I mean, we don't really call it as, well, you can call it a zygote, but this can really be a seed. And especially in watermelon, what happens is that these seeds are harvested from the crossing of a diploid plant and a tetraploid plant, and the seeds are collected and they are planted and they give growth to watermelon plants that are all triploid. With bananas, it's a little bit different. Like they will take the seed, they will grow a banana, and then future bananas are grown from kind of cuttings or shoots from the banana. But either way, the plants are 3n. And that makes it a little bit tricky because if you were to take a 3n or triploid plant, so triploid, and you were to make a sperm and egg from it, you can't, because that is an odd number of chromosomes. So you can't end up with 2n in your sperm and 1n in your egg. Mitosis will fail. And so you would actually never get any seeds being created. And that's why all the bananas that you eat, you're not eating on seeds. Like the regular diploid banana has large black seeds in it. It's kind of disgusting. And that's how you get seedless watermelon as well. It's not a problem if the plant is 4n. You could make 2n sperm and 2n egg. That's easy. If you had 8, any even number of chromosomes is going to get you sperm and egg. Bananas are actually infected with a type of fungus, something called a Cavendish banana. And the Cavendish is ravaged by the fungus. It's destroying huge swaths of crops. And we don't have, well, currently we don't have a defense to the fungus. We have to go about breeding a whole nother line of 3n banana to do this. And so we have to take another type of banana, we have to make it tetraploid, and we have to cross it with a diploid plant. So that's kind of a real life example of why we care about the cell cycle and how we can make commercial fruit larger.